Hello and welcome everyone. I'm Andy Rohrwasser from the Myotonic Dystrophy Foundation, and it's our great pleasure to welcome you to one of our regular Meet the Drug Developer webinars. So today we are very, very fortunate. We are hosting PepGen, and we are going to learn about their progress with their clinical development programs and clinical trials. A little bit about the Myotonic Dystrophy Foundation. So we envision a world with treatments and a cure for the disease. Our mission, or the mission of the foundation, is community care and cure. So we support and connect the myotonic dystrophy community. We provide resources and advocate for care, and we aim to accelerate research towards treatments and a cure. We provide many resources and support for the community. So on our website we have toolkits and publications. On the website you also find information about our support programs. Most importantly, the site also hosts the calendar of events, so where you see what's going on in the community and webinars like today. And then we host the Digital Academy, where you can, at your own leisure, digest webinars and presentations. And hopefully by sometime next week, a recording of today's webinars will also be available there. Many of you have seen this, so please save the date. May 2nd, 3rd, and 4th for our annual conference in Indianapolis, Indiana. We are very excited. We are working on putting the program together, and without giving away any details, it will be a very exciting and informative conference, and we hope to see you all there. I already mentioned it through our website. Please sign up for the Meet the DM Drug Developer webinar series. It's important. You can submit questions. And like I stated in the introduction, today we host PepGen, and then in October we will hear from Arthex Biotech. So this slide is really the most important part of the presentation for me. It represents the myotonic drug development pipeline, and this is really an objective indicator of progress and hope. You see here that we have over 20 commercial programs, and most excitingly, 9 of those are in the clinical testing phase. We update this pipeline regularly, and of course you can find that also on our website. So clinical trial activity falls into 4 broad areas of biology. Going from left to right, we start with the DNA, and the DNA, of course, harbors our gene. In this case, for DM1, the expanded DMPK gene with a repeat. Through a process called transcription, we make RNA, and in DM1, the repeat is also present in the RNA. Because of this repeat, instead of normally processing and folding, we have misfolding, and we form these toxic hairpin structures. And they act like ginormous traps. So they recruit and bind important cellular protein, and they are now not available to do their job in the cell. And the end effect is cellular dysfunction. So in the first category of drug development, we target the DNA. So through inhibition of transcription and through gene editing and gene tuning. The next category focuses on the inactivation of these toxic RNA. The third category focuses on aiming to compensate for these trap proteins. And then the fourth category aims to globally correct cellular dysfunction further downstream. So we have most activity next in category 2, 3, and 4. And PepGen's activity focuses on the inactivation of toxic RNA. So today we are very fortunate. We have a we have a stellar group of presenters. So we have Dr. Jane Larkindale. She's the Vice President of Clinical Services at PepGen, and while she's not presenting, she will be there in the Q&A section to help answering all of your questions. So she has dedicated the last 15 years or so accelerating therapy development in neuromuscular diseases, so supporting research, collaboration, developing data sharing platforms, and assisting with regulatory acceptance of disease model, outcome assessments, and biomarkers. And she has also worked with world leaders in the rare disease space, including the Muscular Dystrophy Association, the Friedreich's Ataxia Research Alliance, the Critical Path Institutes, and of course with our foundation. Then we hear from Alayna Tress. She's the Director of Patient Advocacy at PepGen. So she studied economics and public health, and she loves to learn how patients and family and physicians make healthcare decisions. So her mantra is really that an educated patient is an empowered patient, and that transcends in her professional and private life. So she has 12 years of experience in biotech, with a focus in medical affairs and patient advocacy. And before joining PepGen, she worked on other neuromuscular diseases. And to go a little bit off script to show, right, that this reflects also her personal dedication. So Alayna made a donation to MDF on behalf of her local yoga studio, where she's learning to become a yoga teacher. And she taught a donation-based class in support of myotonic dystrophy in motion and made this donation. So that was just great. Next, we'll hear from Dr. Johanna Hamel. So Johanna studied medicine in Germany, and then she completed residency and fellowship training at the University of Rochester. And her focus and interests are neuromuscular medicine and myotonic dystrophy. As a clinical researcher, she focuses on natural history study, disease progression, biomarkers, and transcriptional changes. And as many of you know, she's also the co-PI of the Wellstone Muscular Dystrophy Specialized Research Center. And then from a patient perspective, right, in the clinic, she cares for DM patients. So Johanna also focuses on remote patient assessment methods and patient registries. And with MDF support, Johanna was able, right, to launch the REACH-DM study. So she really sees these methodologies as a foundational cornerstone for expanded studies of genetic modifiers and real-world impact of new drugs. So before we hand it over to the PepGen team, a little bit of housekeeping. Please ask questions live. Submit them through the Q&A section. We try to compile them and try to answer as many of them as possible. And thank you all for those who already have submitted questions. I think we will have a really nice and robust discussion afterwards. So, without further ado, welcome PepGen team, and we are looking forward to your presentation.
Thank you, Andy, and thank you everyone for making it here today. I'm just going to open up my screen here to kick off the presentation. So today's presentation will be about PGN-EDO-DM1 for myotonic dystrophy type 1. That is the investigational medicine we're studying for the potential treatment of DM1. I want to start off by reminding everyone of PepGen's mission. And our mission is that we're committed to transforming the lives of people with severe neuromuscular and neurological disorders with our enhanced delivery oligonucleotides. That's a mouthful, but you'll hear myself and Dr. Hamel and Jane refer to these throughout the rest of the presentation as EDOs. What we'll go through today is first, I'll walk you through a little bit so far about our commitment to the community and what we've learned from the community so far. And then I'll pass it over to Dr. Johanna Hamel to go through a little bit more in depth about what are our EDOs and how do they work in DM1, what we've seen with them in our non-clinical program and how that wraps into our current clinical study. And then more details about that current clinical study, which is otherwise known as Freedom DM1. So to kick off on how much we've learned from the community so far, we really wouldn't be here today, the stage that PepGen is in with having an open clinical trial, if it wasn't for the community. And that also includes the MDF for all of the insights they've helped us gather. And these are just some of the many examples of insights we've gathered so far. So one, we've been able to have really candid conversations with community members about what we should measure in our clinical trials, what symptoms matter the most to you, what's most uncomfortable, what is your greatest unmet need, and how is that something we can see we can move the needle on or not. Also, what a clinical visit looks like if someone participates in one of our clinical trials. We know that joining a clinical trial takes a lot of time, it takes a lot of effort. So really learning about anything we can do in the scheduling and the order of assessments for when someone comes to a clinical visit for a clinical trial to make it less burdensome on them and the people that they might be traveling with. And then leading into that, Dr. Hamel will go through this in more detail, but we've also gotten a lot of good feedback from the community on how PepGen can best support people who participate in our clinical trials regarding travel services, so booking travel, scheduling travel, liaising with the clinic, and reimbursement for that travel, in addition to other incidentals that may occur. And then the other bullet here, and part of this is why we're here today, is the materials we put together. So we really want to make sure that the community understands who we are at PepGen, what our technology is, and what we're studying. So the community can be better informed about the different options out there and what might work best for them. And then last but not least, the entire drug development process. I know a lot of today we'll be talking a lot about our clinical trial and what that participation looks like. But that is just one piece of a very big and long journey that PepGen and many others have been on. And we really are so thankful for all the advice we've gotten to where we are today. And I'm sure all the advice yet to come for where we need to go tomorrow and beyond. And some of the examples of how we have gathered these insights and continue to gather these insights and engagement. One, we always love in-person engagements. This is actually a photo of one of our laboratory scientists, Colleen, with Janine, who many of you might know as an MDF support group leader. She was able to come to our office, take a tour, meet with staff, and share her story. And I think that was really meaningful to a lot of people at PepGen who don't always have the privilege of being able to engage directly with the community. Secondly, my team, the patient advocacy team, manages an inbox, which is community@pepgen.com. It's really important that we have an open line of communication, and that's something that we monitor every day just to see if anyone has any questions for us or any feedback to give. This middle box here is an example of one of the pieces of educational materials we put out in the past. Whenever we design those, which I feel like we're always kind of redesigning as we get more data, as we get more community feedback on how to explain ourselves and just further better focus our education. It's important that we get your feedback so we know that we're communicating to you in a way that's meaningful and in a way that enables you to be empowered. And then up on the right-hand side here, I think this spring was a lot of fun and very busy for a lot of us at the regional MDF conferences. It was really just such a privilege and so fun just to meet so many of you, across our table, whether it was at a booth or next to you at a table, if it was at lunch. That was something we were really thankful for. And I know all of us at PepGen are really excited for the conference next year, just anytime we can get that in-person engagement with the community. And then last but not least, our advisory boards and patient councils. We've been able to have some wonderful discussions about draft protocols, synopsis, some of our informed consent forms, and other educational materials that are in draft, just to really hear from the community what matters most to them. And we're forever grateful for all that information given to us. And so with that, I'm going to stop sharing my screen and pass it on to Dr. Johanna Hamel, who will take us through the remainder of the presentation.
Okay, very good. Thank you. All right, then I'll just turn on my laser pointer. All right, we're all set. So thank you for having us today. Thank you for spending the afternoon with us and learning more about the PepGen program. And just as a reminder, because I wear different hats, I'm here today because I'm the principal investigator of the study Freedom DM1 that I will be talking about. But before we talk about the study and how you can learn more about this study, we will talk a little bit about the PGN EDO-DM1 technology and the mechanism, and also about the preclinical data and what that entails. We'll get there. And then lastly, we'll finish talking about the trial itself. These are my disclosures. I serve on the clinical advisory board for Vertex and Dyne Therapeutics. And then, as I mentioned, I'm the study principal investigator for Freedom DM1. So now let's dive right in and talk about the challenge. We know that oligonucleotides, these kind of class of drugs, can or have a potential to be very effective in myotonic dystrophy. But the problem has been to get it into the muscle, into the structure where it needs to engage with the root cause of the disease and tackle it. And so to get it in the right place, that has been the challenge. Now, what is PepGen doing to overcome that? One way to think about that is what is the barrier, for example, similar to if you try to get on a Zoom meeting, but you don't have a passcode. There's no way to get in. So what is the passcode, and what kind of passcode did PepGen develop to get into the right place? The solution that PepGen came up with is to link this active component, the oligonucleotide, with a peptide. And through this linker, the drug can enter the cell, is taken up into the cell, and then can go into the nucleus where it really needs to go to help tackle the mechanism in myotonic dystrophy and get there in the right amounts. And so this is really the way to overcome the challenge that's been in the field for many years. And that's one strategy, and that's what you will be hearing about today. So once it is in the nucleus, how does it work? Well, some of the mechanism in myotonic dystrophy you just reviewed with Andy, but I'll go through it again because it is kind of complicated. But let's take a look at the myotonic dystrophy gene RNA, which you see here on the left-hand side. So RNA are molecules that serve as templates to make proteins. And people living with myotonic dystrophy, both types, type 1 and type 2, have a high number of these repeats on this molecule. So in terms of the RNA, at the RNA level, there's too many CUG repeats. And what happens is, because there's a high number of repeats, they form these clumps. And the clumps, here you see that clump, and these clumps act like sponges, and they soak up important RNA binding proteins. We will refer to them as MBNL proteins. Those RNA binding proteins are usually supposed to float around freely in the nucleus and do their jobs, but instead they're stuck here in these RNA clumps. And because they're stuck there in that sponge, they don't do their job anymore. They're stuck and not functioning properly. And so what happens is, well, you might ask, well, what is their job? What are they supposed to do? Well, these MBNL proteins are supposed to edit or splice RNAs from numerous other genes. So for example, these MBNL proteins, they usually splice chloride channel RNA. And because they're not doing it properly anymore, people can experience myotonia. These MBNL proteins, they don't splice properly calcium channel RNAs anymore, and that results in weakness. These MBNL proteins, they're not splicing properly the insulin receptor gene RNA anymore, and that can result in diabetes. And so I could go on, but that results in many problems in myotonic dystrophy and this multisystemic disease that infects different organ systems. And so that's really the root cause of the disease. And because that sponge is causing this issue where these proteins are stuck and the result is misprocessed and misspliced proteins and RNAs, because that's the case, and this is really so toxic, we call it a toxic RNA or toxic foci or toxic clumps. And so what can we do about this here, and how can we rescue this? And that's what PepGen is trying to tackle by developing this PGN-EDO-DM1, which binds to this toxic clump and blocks off these proteins. And by blocking off these proteins, they get released or freed. And now they can float around again in the nucleus and do their job. And the theory is that they do their job, close to normal or much better. And so that there will be normal spliced RNA and normal proteins, and hopefully improve the symptoms from myotonic dystrophy. Now, this purpose of this slide is kind of going through it again in words. So I like the visual better, but some people like the words better. But just to sum it up, so people with myotonic dystrophy have too many repeats, too many CUG repeats on their RNA. And that's because there's too many CTG repeats on the DNA, but that gets transcribed into RNA. So it's really our focus is on RNA today. And these high number of repeats are present not just in muscle. I will provide some information and pictures on muscle, but that also is present in other parts of the body. These repeats cause these toxic foci or these clumps, and we call them toxic because they clump up or soak up these important proteins that get stuck. And then these important proteins, the MBNL proteins, don't do their job anymore. And because they're dysfunctional, we see misspliced RNA and misprocessed proteins. And so just to go back for 1 second here, we actually measure the degree of missplicing in your muscle. When we take biopsies, that's why we take biopsies, because we want to see, well, how does the splicing look when someone is not getting treated? And then we want to look again when someone received treatment. Is there a signal, and has the splicing here improved because those MBNL proteins have been freed? And so that's the approach to get a drug into the nucleus using that combination and with a peptide binding, blocking these CRG repeats, freeing that MBNL, and hopefully improving symptoms of myotonic dystrophy. And the goal is to do that not just, in several muscles, but wherever the problem occurs. That would mean in skeletal muscle, which is the muscle in our limbs, the breathing muscles, but also smooth muscle, which is in the GI tract or some female organs. And to give you an example, or also the cardiac conduction system. Now, I will give you a little bit more of a visual impression of preclinical data. And what is preclinical data? That is data that's generated in the lab. And there's a lot of work that goes into drug development before a drug actually hits the door of the clinical research center, and we can test it in people. So I will go through some of the work that has been done beforehand, and only if that work is promising, a drug actually makes it to the clinical research center. And so what happened to PGN-EDO-DM1? So what was done, what PepGen did, was an experiment in the lab, and they took a muscle fiber or muscle cells from a patient, and that was, again, taken into the lab. And then we can look with special microscopes we can look at these muscle fibers and we can see what we talked about earlier. So here you see in blue the nucleus, and then you see these little red dots. And these little red dots are the toxic RNA clumps. And this fiber was treated in the lab, in the dish, with the PGN EDO-DM1. And what you can see is that there is a reduction of clumps. And that was reduced by 54% in that experiment. And next, here you can see that when you stain those same fibers, not for the clumps, but for MBNL proteins, those proteins that we try to free, you can see that up here in the not treated muscle fiber, you can see that here again, the nuclei in the green round things. And then you see that the MBNL proteins are stuck in those green dots here. And then when the fiber was treated with the drug, you could see that that they became unstuck, so to say, and were more freely floating in the nucleus. And when they are freely floating in the nucleus, we think that then they're going to do their normal business and splice and edit RNAs properly again. And so that, whether they're doing their normal business that they're supposed to do, we can measure by measuring the degree of missplicing of RNAs. And so here in this graph, it looks complex, but I'll go through it with you. So here you see the degree of splicing correction to get back to normal. So up here is normal, and down here is abnormal. And you see, so this, for example, is a normal splicing in a cell that doesn't have myotonic dystrophy. And then when you take a cell that has myotonic dystrophy, you see that if you give the drug here at increasing concentrations, that the splicing goes closer back to normal. Now here I will show you again, I think preclinical data is always a little, it's a little bit, even for us clinicians, it's quite removed. We're not spending a lot, as clinical researchers, we don't spend a lot of time in the lab, but it's important to review this in two ways, to assess a little bit, what are the chances of this drug to work, but also safety concerns. And so that's why I'm spending quite a bit of time here on these preclinical data from the lab. And so here I want to show you an experiment that was done with a mouse that has features of myotonic dystrophy. And this mouse was not, on the left-hand side, was not treated with PGN EDO-DM1. And on the right-hand side, it was treated with a drug. And before I turn on this video, which I actually don't know if I can, let me see if I can turn it on if I take off. Yes, I can. Okay. So this mouse on the left-hand side was not treated, but I have to tell you a little bit where to look at. This mouse has myotonia. It doesn't have all features of myotonic dystrophy, but it has some features. And one of them is that it has quite a bit of myotonia. So if you pinch the hind legs, you will see that they kind of stick out. And then as it warms up, the myotonia goes away. And so that's what you will see. So take a look at the hind legs and what happens when it gets pinched. They kind of stick out. It looks like it's just sticking out the hind legs there. And so that's untreated. And over and over, it takes a little bit, and then it warms up, and then the myotonia goes away. And then on the right-hand side, it was treated with the drug, and it gets pinched there, and you can't see really anything. It doesn't seem like the mouse has myotonia. So it's the same. The mouse has the same kind of. The mouse has the same issue as on the left-hand side, but it has been treated, and so you see that the myotonia is improved. So that is an experiment that was done in the lab. And next, this slide should just summarize for you what are the things that are done before a patient is invited into participating in a clinical trial. So a drug is tested, for example, in a cell in the lab. We talked about that. Then animal studies are done where specific animals that have features of myotonic dystrophy are being tested, and they are getting the drug, and then several dosages are tried out, and then also some clinical features are observed, like does the myotonia go away? How long does it take for the myotonia to go away? Things like that. And then eventually, Bigger animals are being given the drug just for safety concerns. They don't have anything to do with myotonic dystrophy, but just to see if they can tolerate the drug and if there are any issues. And this I will go through fairly quickly because this is kind of a summary of what you learn from preclinical assessments. You learn whether it gets into the drug. You learn whether it gets into the muscle. You learn if it's correcting the misplicing, like in the cell I showed you, whether the myotonia gets better for the mouse that has features of myotonic dystrophy, and how long these effects last. And then you learn a little bit, are there any safety issues when you give it to larger animals? And with that, I want to move on into what happens then, because if all of that looks promising and safe enough, then we can take it to the next step and test the drug in a clinical trial. And that is the Freedom DM1 study that's ongoing as we speak. And first of all, before saying anything else about this, I want to reiterate what Alayna said, and that is thanks to the community, because we learn from you a lot, and we also learn from you how to better design trials and what to pay attention more and how we can reduce the burden. So, for example, talking about inclusion and exclusion criteria, those are criteria someone needs to meet before they can participate and get dosed in a trial, for example. Or, how many assessments should someone do in a day? How fatigued do people get? And we might not get the best measurements if someone feels really fatigued and worn out. And then what symptoms actually matter the most? And helping us to understand, reduce the burden, because it takes a lot of time and effort to participate in a clinical trial. So what is Freedom DM1? It's a phase 1 study to explore a single intravenous dose of the drug, PGN EDO DM1, and it's testing whether it's safe and tolerable for people with DM1 compared to a placebo. And a placebo is a substance that has no active drug. So, for example, it could be a normal saline infusion. So just to summarize, phase 1 are usually early trials where we really focus on safety, where we also are interested in learning what is the best dose. But while we're looking at safety primarily, we also measure some effects on strength and function. The goal is to enroll 24 people with DM1 across the US, Canada, and UK. The randomization is 3-1. So randomized means that someone is assigned by chance whether they receive active drug, or placebo, so the non-active component. And it's a little in favor to get drug because 75% receive drug and 25% receive placebo. So for example, here 6 participants would receive drug and 2 would get placebo. The study timeline is that you first undergo a screening period where several measurements are taken, blood tests are taken, just to make sure it's really, not unsafe for someone to participate in a trial based on those assessments. And that can be up to 30 days. And then once that's passed and someone meets the criteria, the treatment period starts, and that lasts for 16 weeks. It's 1 dose, and then the rest of the weeks is observation. What kind of assessments are done? A variety of things. So we measure, of course, vitals. We test the urine. We have blood draws. The heart gets assessed. We ask you a lot of questions about your medical history, about symptoms. You fill out questionnaires about how you feel and function in day-to-day life. There are strength and motor function activities, and there are 3 muscle biopsies with the goal to look at this splicing issue and whether there has been a change. So that 1 biopsy is done before someone receives study drug or placebo, and then 2 biopsies are done after that. And so what are the criteria? This is not a complete list of inclusion or exclusion criteria, but just some examples. people can participate if they're between 18 and 50 years of age. They need to have a diagnosis of myotonic dystrophy. They must meet a certain degree of strength in both legs as measured by the physical therapists at the screening visit. And that mainly means that people have to have the ability to lift up their toes and cock back their ankle against gravity, and then also be able to resist some moderate pressure when you put your hand on the ankle. And then people need to have hand or grip myotonia, that stiffness of the hand muscles. People cannot participate if they had their symptoms at birth, so congenital myotonic dystrophy. If they have certain conditions, and that's a review by the study doctor to make sure there are no other diseases that could put someone at greater risk when they participate. And then you cannot be on any anti-myotonia medication. So if people take mixilotine or ranolazine or something else that treats the myotonia, that has to be stopped before the study. And then people need to have some residual. So if there's a certain degree of breathing muscle weakness, that can be an exclusion too, and that's being tested when someone screens. Now, one additional point here to make is that PepGen has paid attention to reduce the burden for participants, and the support for trial participants includes the travel service. And so that eliminates the upfront costs paying for even flights or a hotel or anything. So that is taken away. And then there is also this service also helps to communicate between the participant and the site. There's also additional reference guides that provides information on travel support or reminders or frequently asked questions. There is a possibility to get equipment, like if you have to stay when you're staying in the research center or in the hotel, if needed, there is a way to get special equipment. And then just to be an assistance and resource for communication. These are the current sites. This is work in progress. As we speak, there are additional sites that are becoming activated, but these sites are open and enrolling. So 2 in Canada here, in Ottawa, and in Saguenay, and then 1 in the UK. And then here you see the sites in the US at the moment. So we are up here. So that's me. And then we have Richmond, and we have Boston, and then we have Atlanta down here, and 2 sites in California. Now you would say, what about this middle here? And yes, there will be sites soon too. So keep up to date with the study team and with PepGen moving forward. And then this is the PepGen team. And I would like to thank MDF. I would like to thank all the families and study participants and all the people who are interested in the studies and trials, and all those also who learn that they're not a candidate for this particular trial. Then I really say, stay tuned. Every trial is a little different. Don't get too discouraged. And we're moving as fast as we can to get access for our community. And then also thank you for the community, for the clinical advisors, for the sites. And to learn more about PepGen and the program and what's up in the future, here is some contact information. If you have questions, feel free to email. Also, if you want to stay up to date on sites. And with that, I'll move over to the Q&A session, and I'll stop sharing the slides. Thank you for your attention.
Thank you, Johanna. That was great. Again, please ask questions. We have some excellent questions, and I think we will have a robust discussion now. So if all the presenters can, we'll stop the slideshow for a second. And if all the presenters can turn their cameras on, I think that would be great. So what I'm hoping we can do now, we got many questions, right? Many interesting questions. I try to put them in categories. So maybe category one can be clinical trials, ins and outs, and then we can talk a little bit in the second phase about the biology, and then we can maybe at the end get the crystal balls from Johanna and from Jane with regards to where they see this will all go. So maybe you can start. Maybe that's for Alayna and Jane. Clinical trial sites, additional clinical trial sites. What needs to happen? What can we do? Because as one of the attendees correctly points out, right? If we cover the middle of the United States, we reduce travel burden, and we have a much greater patient population.
Yeah, we obviously really recognize the fact that travel's not easy for anyone, let alone if you're living with myotonic dystrophy. The sites that Johanna showed are our currently open sites. We have other sites that we are working to get open, really particularly filling that donut hole in the middle of the country, and those will be coming online hopefully in the next few months. Obviously, we can't have sites everywhere in the world. We need to have some control over both how many places we're sending our drug to, and just in general how many sites we have open. So we're trying to optimize making sure people don't have to travel too far, while at the same time making sure that we have our study being done at high quality sites, well distributed sites, places where patients are being seen already. I know, and this is a topic very close to Dr. Hamel's heart, is one of the things that's of great interest is remote assessment and having people travel for fewer sites. It's unusual to use those in early stage studies because you want to assess the safety very closely in the early stage studies, but it is something we are interested in and want to learn more about as we move to larger and later stage studies. And Dr. Hamel, I don't know if you want to comment about that aspect.
Yeah, I think I don't know. I can just emphasize what you said. We're working hard to, make sure we're ready for the future, to reduce the burden, and once there are drugs out there that we can test them at a reduced burden for people in the real world. But phase 1, phase 2, those kind of trials, really safety, getting and evaluating people in person on site, getting those blood drawn, that's really key. And so, yes, but I think we'll move towards that where we can reach more of the community, and that will be in the future, but not quite yet.
So maybe Jane, to put you on the spot again, so patients here, right? What's the message for patients? A patient who wants to participate in a trial, give them the step 1, 2, 3, so to speak.
Yeah, so the simple answer is yes, we'd love to have you take part in the study. The first thing, if you don't know anything, look at clinicaltrials.gov. The website was on the last slide and we can share it, or email clinicaltrials at pepgen.com. That will get to Alayna. She will answer you. We can guarantee she will answer you. From there, you can identify where the closest site is. And if you're lucky, there's a site very close to you and you can call up the coordinator and see if you can come in. If there isn't, call the coordinator of somewhere you want to go that's reasonably close. We do cover travel. We try and make it as easy as possible, arrange your travel for you so you can come into the site. So we're going to do everything we can to make it possible, even if the site isn't particularly close to you, and try and support you in coming in. The third step, once you've identified a site, they've called to talk to you, you come in, the clinician there will have to assess you for those inclusion-exclusion criteria that Dr. Hamel spoke about. Unfortunately, particularly in early study trials, not everyone can participate. We're typically looking at a fairly tight population so we can really understand safety and how the drug works. So unfortunately, not everyone can take part at this point. As development goes on, we may open those requirements up. So in future studies, we may have slightly different criteria, and every study has its own criteria. But then, if you do meet the requirements, then you can come in and be a part of the trial if you want to. It's always up to you. There is no requirement to do this. You can go all the way up to that point. They say you can be a part of the trial, and if you decide you don't want to, that will always be your choice.
Very good. So to put you on the spot again, the next two questions are pertaining to timelines. So in an ideal world, and the world is hardly ever ideal, but if anything goes to according to plan, could you line out the next, so the timeline for the conclusion of all the clinical testing phases, right? And then the second part of the question would be the timeline, if we had an effective drug, how long it would take to come to market.
Those are very difficult questions because, as you say, the world is not ideal. What I can speak to is the current trial is ongoing. It will continue to be ongoing for at least into the next year. At the same time, because we are very optimistic about this therapy, we think our non-clinical results are very promising. We are starting a multiple ascending dose study, the next stage of study. I believe in our recent guidance, we're hoping to get that open by the end of the year. So that will start even before we finish the current study, because we're really trying to accelerate development here. So those are the things I can say. Then how long will it take after that? Those are still ongoing conversations as to exactly what they call a pivotal study will be, the last study you need before approval. So I can't really give any guidance on how long that will take until we have a design that we've agreed with the regulators. But typically those will take at least a couple of years beyond that. And then you have to get approval here in the US by the FDA, in Canada by Health Canada, MHRA in the UK, EMA in Europe and such like, and all of that quite rightly takes some time while the regulators really review all the data, safety, efficacy, all the data from every trial. And it takes them some time before they can say, yes, this drug is okay to be used by patients, by any doctor at any time. So all of that is a fairly lengthy process. So we'd be talking several years at this point.
And then maybe your perspective, right, based on your experience in this space, if you could also comment on there were questions with regards to pricing and availability, how that works in very general terms. We know this is very far away, so.
Yeah, it's very far away. And to be honest, I have not been part of many of those discussions. Obviously, we all recognize that rare disease drugs are often quite expensive. Because simply put, it takes a lot of work from a lot of people to get a drug all the way through development, and a lot of drugs don't work along the way. So I can't really speak specifically towards pricing of our drug, but it's something we're very conscious of, that we know that these tend to be very expensive, and you have to get those appropriate regulatory approvals so you can get insurance cover, at least here in the US.
Great, thank you. So the next set of questions, let's transition into a little bit into biology, and there's actually a good question in terms of maybe if you could comment on dosing, how you figure out the lowest effective dose, right, or the maximum dose necessary to be clinically effective. Again, maybe general, maybe give a general answer how this is done.
Yeah, so Dr. Hamel referred to the huge amount of non-clinical work that's done before a drug even gets to the clinic. We spend a lot of time looking at different doses in different animal models, and mostly mice and non-human primates. And we're really looking there, what level are we seeing some of the biological changes we want to see? We're in the models where we can measure them. What levels of drug are we measuring in the tissues? Where is the drug going? How frequently do I have to dose to see those changes that we want to see? In the case of myotonic dystrophy, where we don't have particularly good models, an awful lot of this is based on how much drug we get into different tissues. The mouse model we use only really has symptoms in the muscles, so it's not very useful for ascertaining if you're having an effect on the heart, say. But we know that the drug gets there. So at least we know how much drug gets there with what dose. And then we extrapolate that to humans before we go into the study. And this is why clinical trials tend to start at a low dose, which is way below what we've looked at in animals typically, and then slowly go up as we look at the safety data, look at those drug levels, look at any efficacy, and see if what we're predicting based on the animals is actually going to match forwards to humans and ditto the safety.
Thank you. So Now, Johanna, and again, Jane, your perspective on, so let's say this drug is effective. We always talk about this is always the focus here is DM1. Do you see, what's your perspective on efficacy in effectiveness for DM2 patients?
I can say maybe one brief comment in, because I do see a lot of patients with DM2, and it's always not easy to talk about the advances in DM1 and then not being able to provide a similar story yet for DM2. But the way I would usually phrase it is that not all approaches, tested at the moment in the community for DM1 are applicable to DM2, because we cannot just knock out or silence or reduce CNBP, the DM2 gene. That RNA is probably needed. But thinking about, as a clinician, thinking about the PepGen approach, for example, it's not really knocking down the gene, it's just rescuing those proteins, releasing those proteins. And so that is something of interest for me as a DM2 clinician. And then what I also say to my DM2 community is that we learn a lot beyond the particular drug and what happens exactly in DM1, but we learn about outcome measures, we learn about safety, we learn about trial design, and some of that doesn't have to, probably I'm hoping, doesn't have to be reinvented for DM2. When we get there, I think we'll have a lot more knowledge.
Yeah, and I guess the only thing I can add to that really great answer is in the case of PGN EDODM1, the target of the oligonucleotide, as we explained earlier, is really the repeat sequence. The repeat sequence for DM2 is not identical to DM1. So unfortunately, PGN EDODM1 would not work in DM2 patients. But having said that, a similar approach absolutely could, and it's something we're interested in, is assessing whether a similar approach could work in DM2, but obviously not the same drug and much earlier.
Great, thank you. So let's go a little bit after the so maybe again both of your opinions in terms of, let's assume the drug is effective, and considering the myriad of symptoms, right, of the disease, where would you see, maybe Johanna, you first, as a clinician, where would you expect to see or hope to see the biggest effect of the drug would manifest first?
Yeah, we'll see. I don't know. That's why we're testing it, and I can always be surprised. I've learned so much in the last 3 years about myotonic dystrophy by doing trials. So I have an open mind. But that being said, I think we all have a sense that myotonia is probably quite responsive. And if the community has observed that we spend a lot of time talking about myotonia and showing, even mice that recover from myotonia, that is because it seems to respond quickly, and it might give us an idea quickly whether something is working or not, or whether at least it does in the tissue what it's supposed to do, and does something very myotonic specific, reducing myotonia. However, that by no means does that mean, okay, we're done, great, myotonia is gone, because we all know that for some people it's the most severe and bothersome symptom, but for a lot of people it's not. It's actually ranks quite low on the symptom priority list. That doesn't mean that the drug won't work on other tissues and really, but I think it might take longer. That's my clinical sense. It might take longer to show these other effects, while myotonia might be a quicker response. That's how I look at it.
Jane, do you want to add anything here, or maybe from the perspective of how would you capture or assess non-muscular improvement?
Yeah, it's a really interesting question. And one I think I've spent the last 2.5 years really exploring is what other symptoms can we measure, when are they likely to change? And as Dr. Hamel said, I think we're learning all the time. Every time a study reads out, we learn more. What we know about EDODM1 is that it gets into a lot of different tissues, so it has the potential to affect lots of different symptoms. And really, it's one of the things we've explored a lot with clinical experts and also with the community is how many assessments can we do? Which ones are actually going to be meaningful? When are things likely to change? So obviously, I think that we're going to look at myotonia because as Johanna says, I think that's probably one of the quickest to change. But we're also interested in muscle strength, ability to walk, some of the muscle function things, looking at the quality of muscle. One thing we've heard loud and clear from the community is the importance of some of the GI and swallowing issues, and we're exploring some swallowing endpoints as well, because those are obviously very important measures to the community. What we haven't found is great ways to measure some of the cognitive or fatigue aspects of disease that are also very clinically meaningful, or some of the cardiac aspects, and those are still things we're looking into. It's something I'm very, very interested in, but I just am not aware of good, high quality assessments that would have any confidence in what we would measure. So it's not because we don't think those symptoms are important. I know they're important, but they're very difficult to measure. So there's always a balance in a study. We don't want to give you so many assessments. You've got to be at the study site for 10 days. We don't want to do assessments that are going to be so fuzzy we can't reinterpret the results. But we do want to measure things that matter to people. I don't care if your left pinky finger just stops hurting. It's probably not the most important thing in your life. So we're balancing all of those things.
Great. So maybe the next question is really sciencey and almost geeky, in terms of maybe can you explain so when the mutant DMPK RNA is freed of the sponges, what do you think happens with it?
Yeah, and what we've shared publicly, that the one thing we do know for sure is the level of DMPK doesn't change, at least not in non-clinical models. So we don't believe it's degraded. Beyond that, we don't know. Presumably, if it's not being degraded, it's still going about whatever its function is. We're not actually a scientist 100% sure what that is. So a whole lot of I don't know, but we do know it's not degraded.
Great. So looking at my list and looking at the list of submitted question, I think we probably answered all of them, right? So what I want to use, I would like to first of all thank you. That was super helpful and I think hopefully really good for the community. I would like to transition now to the to make a plea to participate in registries, because as we hear, right, as Jane said it, right, that maybe the next phase of clinical trials is to assess cognitive impact and non-muscular impact, and for that we need more trials. And on top of that, we have more of these novel entities that will hit the clinic. So please participate in registries. It's really, really important. It's the it's really the foundational piece, right, towards finding this cure. So again, a big thank you to the PepGen team. It was fabulous, and hopefully many of you will look at the recording. Clete, can we go back to the can we go back now to the closing slide? So with this thank you to the PepGen team. Now for our combined audience. So if this webinar was helpful and if it did speak to you, please consider, right, showing your appreciation with a donation to MDF. And for those affected by the disease, please participate in the registry. It really is a foundational piece for the future, and only if we do this, only together we can make change, right, and find this cure. So with this, I would like to end. I would like to thank PepGen one more time, and Johanna one more time, and all of you who attended the webinar, and all of you who submitted questions, and hopefully revisit the recording and found benefit in this presentation. And with this, I would like to end. Thank you again, and until next time.